Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad PhD Entrance Examination question paper for the subject Biotechnology, and I will be discussing only the biology part. The exam was conducted in the year 2019. So coming to the first question, supercoiled DNA is soluble in. So it is soluble in water saturated phenol. Answer is option B. Then identify the mismatch from the following. Solubilization of the membrane lipid bilayer SDS. Digestion of prokaryotic cell wall lysozyme. That's also correct. Cleavage of ester bond in diacyl glycerol phosphate. That is by lipase. That is also correct. Digestion of RNA of DNA RNA hybrid RNase A. It is not RNase A, it is RNase H. So the mismatch is option D. Then next one. Identify the correct match from the following combination. Real-time PCR expression profiling of all the genes at a time in an organism. Real-time PCR in real-time PCR, the accumulation of amplification product is measured as the reaction progresses in real time with product quantification at each cycle. So this is the incorrect mismatch. It is not expression profiling of all the genes of an organism at the same time. It is meant for quantitative analysis of the reaction products. Okay, not for expression profiling of all genes at a time in an organism. Next one. You are extracting DNA from Saccharomyces cerevisiae. If in your final spin you discover that you have large white pellet. You dissolve the pellet in TE buffer, then use the spectrophotometer and get an A260 by 280 ratio of 1.4. Which of the following best explains your observation? So here you are doing the process of isolation of DNA from Saccharomyces cerevisiae. You have got the final spin. You have come to that stage and you have got a large white pellet. Then you dissolve the pellet in TE buffer. The large white pellet is with DNA. Okay, so you dissolve the pellet in TE buffer and then you take spectrophotometer reading, that is A260 by 280 ratio. This is for knowing the purity of your sample. But you got a ratio of 1.4. So if it is 1.8, then it is pure DNA, and if it is approximately around 2, then it is pure RNA. So here you got the ratio as 1.4, which shows that DNA is contaminated with proteins. Answer is option C. Then next one. You have isolated a bacterium that exhibits very poor growth in the presence of low tryptophan. You then hypothesize that a mutation occurred in the TRP repressor, causing it to bind too strongly to the operator. Which of the following experiments would best confirm your hypothesis? So first option is sequence the TRP repressor, perform a clustral W alignment to compare the new sequence with the wild type sequence, and then perform an EMSA, that is gel mobility shift assay. So that option is correct. Why? Because clustral W, it is used for multiple sequence alignment. So once you sequence the tryptophan repressor, you can perform the clustral W alignment, and through sequence alignment you will know if there is any mutation in the tryptophan repressor. Then you have to perform the EMSA method. This EMSA is for detecting protein DNA interaction. If protein is mutated, it will not bind to the DNA. Otherwise, it will bind. So from these experiments, you can confirm your hypothesis that a mutation occurred in the TRP repressor, causing it to bind too strongly to the operator. So the answer is option A. Then next one. A survey of numerous bacterial species was done to identify in each case the sequence recognized by the endogenous DAM methylase. Using synthetic DNA fragments, gel mobility shift assays and direct assays for the methylation of DNA, 4 new sequences are identified from different organisms that are recognized by the DAM enzymes. In one of these bacteria, the DAM methylase is not used to mark newly synthesized DNA for mismatch repair. Mark the sequence. These sequences are. So what they have done is a survey of bacterial species was done for what? To identify in each case the sequence recognized by the endogenous DAM methylase. Now what does DAM methylase do? It adds a methyl group to the adenine of the sequence 5' GATC in the newly synthesized DNA. That is what DAM methylase does. So using synthetic DNA fragments, gel mobility shift assays and direct assays for the methylation of the DNA, 4 new sequences are identified from different organisms that are recognized by the DAM enzymes. Then in one of these bacteria, the DAM methylase is not used to mark newly synthesized DNA for mismatch repair. So in one of these from one among these numerous bacterial species which they have chosen for experimental work, one of these bacteria, the DAM methylase is not used to mark newly synthesized DNA. So in one of these, the DAM methylase is not marking the newly synthesized DNA. I already told that DAM methylase is an enzyme that adds methyl group to the adenine of the sequence GATC in the newly synthesized DNA. So in one of these bacteria, the DAM methylase is not marking the newly synthesized DNA for mismatch repair. So mark that sequence which is mutated they have asked. So these sequences are they have given. So this is the DNA. This is the DAM methylase added methyl group to the adenine of the sequence GATC, and upon replication, one half will have the methyl group like this. Then the other group, the newly strands are methylated. Then these strands, the newly formed strands, they are also methylated by the DAM methylase. So it methylates at the sequence GATC, that is to the adenine of the sequence GATC, it methylates. So out of these, if you see the first one it has got a GATC, so it is not mutated, it is a normal bacterial sequence. Then here it is CATG. Okay, so there is no GATC in that. Here GTAC is given. So GTAC is present instead of GATC. And here also in the fourth sequence, GATC is not present. So the answer will be option C, that is GTAC. So that is instead of A, there is a T present. So that will be the mutated version. Then next one. A typical microRNA is expressed in neurons so that it is abundant during the night but absent during the day. So microRNA that is expressed in neurons, it is such that it is abundant during the night but absent during the day. Which of the following statements would be unexpected based on this information? Now before coming to the options, microRNAs are class of small non-coding RNAs of 22 nucleotides in length which are involved in the regulation of gene expression at the post-transcription level. And what does it do? It degrades their target mRNA, thereby inhibiting their translation. And microRNAs, they are only partially complementary to the mRNA, not 100% base complementarity is there, it is only partially complementary. And the next term is P bodies. These play fundamental role in mRNA decay. Then the next term I would say is RISC. RISC is RNA induced silencing complex. It is a multi-protein complex that incorporates one strand of a small interfering RNA or microRNA. And these RISC, they use siRNA or mRNA as template for recognizing complementary strand, then it activates RNase and cleaves the RNA. So that is what RISC does. Now what have they told? Option A. Target sequences identical to the miRNA would be expected to have high transcript abundance during the night and lower abundance during the day. So target sequences identical to miRNA. That is, target sequence is your mRNA sequence. That is identical to miRNA. It is not identical to miRNA because it is only partially complementary. So option A is not correct. Then next one, target transcript sequence with less extensive similarity to the miRNA, yes, that sentence is correct, would be expected to have low translation during the night and higher translation during the day. So that is correct, okay, because during the night the mRNA will be degraded because more miRNA is expressed more at the night time. So mRNA will be degraded. So B is correct. Then next one. Target transcript sequences with less extensive similarity to miRNA would be expected to accumulate in P bodies during the night. Of course, because at night miRNA is abundant, it will bind with the less extensive similarity to the mRNA and the mRNA will be targeted to decay. And P bodies, they play a fundamental role in mRNA decay. So option C is also correct. Then next one. Target transcript sequences with less extensive similarity to miRNA would be expected to be associated with risk during the night. Yes, because risk is also involved in RNA degradation. So miRNA is more at night. It will form complex with the RISC and thereby degrading your RNA. So that is also correct. So which is not the correct option? Option A is not correct and that is the answer. So answer is option A. Then next one. In cancer cells, a protein that is normally degraded through the ubiquitin pathway accumulates to high level. What might explain this accumulation of the protein in cancer cell? So before reading the options, the addition of ubiquitin to a substrate protein is called as ubiquitinylation, and ubiquitinylation, it marks the protein for degradation via the proteasome. Then ubiquitinylation, it involves three main steps. First is activation by ubiquitin activating enzymes E1. Then you have got the conjugation, it is by ubiquitin conjugating enzymes E2. And then you have got the ligation being performed, that is ubiquitin ligase E3. So the result of this sequential action of these various enzymes is to bind ubiquitin to the protein targeted for degradation. Now, first option. The amino acid residue that is normally polyubiquitinated on the target protein has been mutated so that it is no longer recognized by the E2. So what they have told is, in cancer cell, protein is normally degraded through the ubiquitin pathway. Not in cancer cells. In normal cells, the protein is normally degraded through the ubiquitin pathway. But in cancer cells, a protein that is normally degraded through ubiquitin pathway is accumulating to high levels. So in cancer cell, the protein that has to be degraded in normal cell is accumulating at high levels in the cancer cell. So that is, in cancer cell, the protein is accumulating at high levels. Now what may be the reason for this high level accumulation of this particular protein? So the first option they have told is, the amino acid residue that is normally polyubiquitinated on the target protein has been mutated so that it no longer recognized by E2. That is one option, because E2 is ubiquitin conjugating enzyme. So if the amino acid residue that is normally polyubiquitinated on the target protein is mutated, then it will not be recognized by the E2. So it will be accumulating at high levels in the cancer cell. So that is one reason. Then next one, the degradation signal on the target protein has been mutated so that it no longer is recognized by E3. So E3 is ubiquitin ligases. So the degradation signal on the target protein. So the degradation signal on the target protein is mutated so that it is not recognized by E3. So that is also another option. Another option C. A mutation has occurred so that E2 and E3 pair that normally ubiquitinate the target protein no longer interact. That may also be a reason. Okay, that these two proteins, that is E2 and E3, they not normally interact. That also can be a reason. And option D is A, B, C could explain the observation. Yes, that is correct. Option D is the answer. Then next one. Where are the synaptic vesicles containing neurotransmitters located? So they are located on the presynaptic end. That is option A. Then next one. Consider the following restriction map below. The solid bar represents a portion of this DNA that has been used as a probe for southern blot. So this bar represents the section of the DNA that has been used as a probe for southern blot. So during southern blot, the probe will bind over to this region. DNA digested with EcoR1 alone is separated and subjected to a southern blot using the region of DNA indicated by the solid bar. So EcoR1 digestion means this region it will be lost, as well as here, okay, if there is an extension of this DNA over here, that region also will be lost, and only this particular region will be remaining. So the probe will bind over there. The size of the bands seen on the southern blot can be represented as. So what will be the size of the band that you observe on your southern blot? So here this portion will be lost. So what is remaining? 1 plus 0.8 plus 0.9, and that you will get it around as 2.7 kb because that will be detected by the probe. So the answer is option C. Then next one, apoptosis is a form of cell death. Answer is option A. Memory loss is not associated with. So memory loss is not associated with amyotrophic lateral sclerosis. So in sclerosis, it is progressive nervous system disease that affects nerve cells in the brain and spinal cord, causing loss of muscle control. It affects mainly the motor neuron. And it is a motor neuron disease. So memory loss is not associated with amyotrophic lateral sclerosis. Next one. Which of the interaction between T cell and APC? APC is antigen presenting cells would lead to energy. So energy in T cells. So TH cell recognition of an antigenic peptide MHC complex. So TH cell, it will recognize an antigen that is presented on MHC complex. So a T cell recognition of an antigenic peptide MHC complex sometimes result in a state of non-responsiveness called clonal energy. So in some cases the T cell which is bound to the antigenic peptide carried on an MHC complex, it doesn't initiate any immune response. It is in a state of non-responsiveness and it is called as clonal energy. So which of the interaction? So engagement of the inhibitory receptor CTL4A by its B7 ligand is critical for the initiation of energy. So the answer here is option C, that is B71 CTLA4. Answer is option C. Then next one. What is the net charge of arginine at pH 1? So if you see the structure of arginine, you have got a COH group at one C terminal end, NH2 group at the N terminal end. The pKa of COH group is 2 and that of NH2 group is 10. Then again you have got a side chain NH2 group with a pKa of 12. So they have asked the net charge of arginine at pH 1. So at pH 1, in the case of carbonyl side, that is C terminal end, pH that is 1 is less than 2 because 2 is the pK value of the C terminal end of this particular amino acid. So there is abundance of protons in the solution at pH 1 and it will be fully protonated. That is the amino acid the C terminal end will be fully protonated and this will give a neutral charge for the carboxyl group. Then you have got the amino groups. That is, one is at the side chain amino group and the other is at the N terminal amino group. So these two combined together will give a net positive charge. So it will be plus 2. Answer is option A. Then next one, the term anomers of glucose refers to. So they are isomers of glucose that differ in configuration at carbon 1. So answer is option D. These are the anomers of glucose. That is alpha D glucose and beta D glucose. So this is the carbon 1. The OH is facing down and here the OH group is facing up. So there is a difference in the configuration at carbon 1. This is the carbon 1 and it is called as anomers. So alpha D glucose and beta D glucose are anomers of glucose. Then next one, match the following. Hard ionization. Hard ionization, electron impact. 1, 3. Then you have got soft ionization. Soft ionization, MALDI. MALDI. Option 4. Mass analyzer is quadrupole and option 1 and the resolution is full width or at half maximum. So answer is option B, that is 1 3 2 4 3 1 and 4 2. Then next one, incorporation of which one of the following nucleotide triphosphate leads to termination of polynucleotide synthesis during Sanger's sequencing reaction. So you know in Sanger sequencing reaction the DDNTP is used as the terminator. So here it is 2' 3' deoxynucleoside triphosphate. That is the DDNTP. Answer is option C. Then next one. Which of the following technique or method is used to determine stoichiometry of interacting proteins? So isothermal titration calorimetry. It is used to measure stoichiometry interaction between proteins. Answer is option C. Then next one. Which of the following radioactive compounds is most commonly used for studying DNA synthesis in cultures of E. coli? So DNA, it contains large quantity of phosphate and there are phosphodiester linkages also. So it will be P32, phosphorus 32, that will be the more commonly used for studying DNA synthesis in cultures of E. coli. So answer is option D. Then next one, in an effort to produce gene knockout mice. So you want to produce a gene knockout mice. A gene targeted homologous recombination. So a gene targeted homologous recombination with exogenous DNA containing neomycin resistance gene that confer G418 resistance and TKHSV gene that confer sensitivity to the cytotoxic nucleotide analog gland cyclovir. If the neomycin resistance gene was inserted within the target gene in the exogenous DNA and considering that both homologous and non-homologous recombination, that is random integration, is taking place, which one of the following statement is not correct about the possible outcome of the experiment? So they want to conduct an experiment where they want to knock out a particular gene in mice. So they have got a exogenous DNA. This is the exogenous DNA. This DNA is having a neomycin resistance gene and a gland cyclovir sensitivity. That I have marked as GS, and neomycin resistance I have marked as neo-R. And what they are doing, They want to knock out this particular gene in mice. So how they are planning to knock out is, you recombine, that is they undergo homologous recombination, and neomycin will come here, and this particular region will become knocked out. Okay, because it is been knocked out by using neomycin resistance gene, because neomycin is going and sitting over to that place. So the gene is already knocked out. So if the neomycin resistance gene was inserted within the target gene in the exogenous DNA, and considering that both homologous and non-homologous recombination is taking place, which of the following statement is not correct about the possible outcome of the experiment? So cells with non-homologous insertion will be sensitive to ganciclovir, of course, because it is non-homologous recombination. It is random integration, so that can happen. So A can be correct. Then non-homologous recombinants will be sensitive towards G418 and resistant to ganciclovir. So non-homologous recombinants, that is they will be sensitive towards G418. So if there is no recombination taking place, there won't be any if homologous recombination is not taking place, then the non-recombinant cells will be sensitive towards G418. Yes, that is here. If you see that it is sensitive towards neomycin resistance, and it will be resistant to ganciclovir. So it will be resistant to ganciclovir. That is your targeted DNA. Okay, not the exogenous DNA that you are using for gene knockout. It is your DNA in the mice that they have asked, which has to be knocked out. That will be resistant to ganciclovir, but it will be sensitive towards G418. Then next one, homologous recombinants will grow in G418 containing media. So this is homologous recombination, and it doesn't involve the use of any restriction enzymes. Okay. Okay, homologous recombinants will grow in G418 medium containing media. So these are your recombinants. Recombinants means they will be ganciclovir resistant as well as neomycin resistant. So homologous recombinants will grow in G418 media. Yes, that is correct. But will be sensitive towards ganciclovir. No, it will be resistant to ganciclovir. Only then we can know that homologous recombination has taken place. Then option C is not correct. So they have asked for which one is not correct. So option C is not correct. Then coming to option D, homologous recombination will ensure that cells will be resistant to both ganciclovir and G418. That is also correct. Through homologous recombination, we will get cells that will be resistant to both ganciclovir and G418. So that is correct. Then next one. Select the correct answer on the basis of the following statements. Continuous rotary vacuum filter press has high labor cost. Continuous rotary vacuum filter press has high clogging. So the main process in rotary vacuum drum filter is continuous filtration, whereby solids are separated from liquids through a filter medium by a vacuum, and it is low cost and no clogging. So here both the statements are false. So answer is option C. Then which of the following approach is associated to the prediction of protein-protein interaction? So it is domain fusion, Rosetta Stone. Answer is option A. Then nanomaterials can be used for chromatography by. So they can be used for chromatography by option D, that is functionalization. And functionalization, it refers to the surface modification of nanoparticles, which includes conjugation of chemicals or biomolecules to the surface, or you can even conjugate them with folic acid, biotin molecules, oligonucleotide peptides and antibodies. So that is called as functionalization. Then next one. Which of the following expression system would not be preferred for obtaining large amount of proteins for crystallization? So it is the case of mammalian cells, that is option B. Why? Because as compared to insect cell, glycosylation in mammalian cells results in attachment of large and complex glycans to the expressed protein which interfere with the crystallization. So the answer is option B, that is mammalian cells. Then next one. Which of the following sequences contain the pattern? So here in this is a prosite pattern. Okay. Prosite. In prosite, this particular square brackets, it means these amino acids, they have either of these has to be present. Either G has to be present or M has to be present. Not both. X is any amino acid is accepted, and this repeated means it can be repeated 3 times. Repetition of an element of the pattern. Okay, then you have got G, H and VT. So we'll see option A. Option A first should be either G or M, but here it is V. So that itself is not correct. Then option B. First has to be G. Okay, G has come. X can be any amino acid and it can be of three residues. So G, L, K, that is correct. Then you have got G over here. Yes. Then H. H is not present. Instead of that, R is present, which is also not correct. Then option C. Again the first amino acid they have written as A instead of G or M. So that is also not correct. And option D, you have got M first. Okay, that's correct. Then V, K, R. Okay. Then G, yes, G over here, H, H and either T or V and it is T. So option D is the correct answer. You can expect question like this which are, and these are very easy to answer. So please learn the prosite pattern. Then next one. A correlation table was obtained for 5 genes from the expression data across 15 samples. So this is the table that they have obtained, correlation table. And what is correlation? It is a degree to which two variables are related to each other. How gene 1 is related to gene 2, that is over here. How is it related? How gene 3 is related to G1 or G2 or and correlation coefficients. They measure the strength between the relationships, that is between the two variables. If the sign on the correlation coefficient represents the if the sign is positive, okay, if the sign for correlation coefficient is positive, then it indicates the value of the variable increases as the value of the other variable increases. And if it is negative, it shows that when the value of one variable increases, the value of other variable tend to decrease. So the extreme values for coefficient is -1 and +1. And a coefficient with 0, it represents no linear relationship. That is gene 1 to 1, it is 1 and it shows a highly perfect correlation between gene 1 and gene 1. So naturally that is correct also because it is with the same gene that you are correlating. So it will naturally be 1. So it shows a higher and very strong correlation. Then, if the correlation coefficient it approaches -1 or 1, the strength of the relationship increases. So it has to be somewhere near the value of 1, then it will be a strong correlation. Okay, now we will see the options. So gene 1 and gene 2, that is gene 1 here and gene 2 over here, it is they have given as 0.894. So 0.894, it is approximately nearer to 1, value 1. So it is a strong correlation. strong positive correlation because the sign is positive over here. So it is strong positive correlation. Then comes gene 4 and gene 1. So that is even more higher value than 0.8. So it is very strong negative correlation because it has got a negative value over here. So as the value of this increases, this one decreases. So it is having a negative correlation. Then next one is gene 5 and gene 3. It is very less than 1. So it will be a very weak positive correlation. Then gene 5 and gene 4, that is over here. It is somewhere in between. So it will be in between the value of 1 and minus 1. So it will be a weak positive correlation. So they have asked which of the following gene pairs show strongest relationship. So it is naturally this one, that is very strong negative correlation. That is 0.962 minus 0.962. So answer is option B. I have given a tabular column for positive and negative correlation for values between 1 and minus 1 for R. R is the coefficient of correlation. You can see it in the next. Okay, it's here. You can just go through this if you want. But just remember that the value closer to 1, it will be more strongly correlated than the value which is further away from 1. The next one. Positive sense RNA of some viruses is used as. So positive sense RNA of some viruses is used as mRNA. Answer is option C. Then tuberculin skin test is an example of. It is an example of type 4 delayed hypersensitivity. Answer is option A. The carbon skeleton are derived from the following molecule for the synthesis of alanine. So it is pyruvate that is used for the synthesis of alanine. The next one. Legumes form a symbiosis with the nodule-inducing bacteria. Identify the substrate provided by the host cell to the bacteroids during this process. So the legumes, they are providing nitrogen fixation, helping the plant with nitrogen fixation. And what does the plant provide to the legume? That is what they have asked. So it is the plant provides malate as source of energy for the bacteroids. Then next one. NADH oxidation is associated with the following reaction during photorespiration. So it is conversion of hydroxypyruvate to glycerate. Answer is option B. Then next one. The cell wall of the following is not rigid in. So it is not rigid in the case of protozoa. Answer is option C. Then next one. The virus that uses reverse transcriptase in its life cycle is. So it is hepatitis B virus, HBV, that uses reverse transcriptase during its replication. So HBV is the answer. Then next one. The disease that can be cured with gene therapy is. So out of these, the disease cystic fibrosis can be cured with gene therapy. Answer is option C. The drug fusidic acid is being used against. So fusidic acid, it is used against Staphylococcus aureus. Answer is option A. So next one. Suppose you have been given the following ROC curve for two different machine learning algorithms A and B. Which of the following algorithm would you choose as final model? So it is the relation between specificity and sensitivity, that is the ROC curve. So if the curve is close to the top left corner, that is over here, I'll show it. If the curve is close to the top left corner, that is over here, that is more towards this corner, then the indicate a better performance. Okay, it indicates a better performance. And if the curve is closer to the 45-degree diagonal, this is the diagonal. If the curve is closer to that diagonal, then it indicates less accurate test. So the answer is which algorithm you would choose as final model is of model A, because it is towards the top left corner and it indicates a better performance. So the answer is option A. Then structure of envelope of on HIV-1 particle. It is trimeric. Answer is option B. Then next one. Activity of topoisomerase 2 not exhibited in the absence of ATP. That is religation. Answer is option C. Then next one. In plants, the abundance of following molecule is noticed during abiotic stress. So which are those molecules? It is ROS, reactive oxygen species, glutathione, oxalic acetic acid, PEP. So the answer here is 1, that is ROS and glutathione are noticed during abiotic stress, that is reactive oxygen species and glutathione. Answer is option C, 1 and 2. Then next one. For a FRET pair, the efficiency of the energy transfer is related to the distance between the pair as. So ET is proportional to R raised to minus 6. That is the answer, option D. Then next one. Dysfunction of dopaminergic neurons associated with the disease. It is Parkinson's disease. Answer is option C. Then which of the following algorithm is used by global alignment? So for global alignment, it is Needleman and Wunsch, and Smith algorithm is for local alignment. Answer is option A. Then which of the following are the literature database? So it is PubMed and Medline. They are literature database. Answer is option B. Then malignant malaria is associated with. So malignant malaria is with Plasmodium falciparum. Answer is option B. Then next one. Which of the following insects belong to the order Lepidoptera? So butterflies, they belong to Lepidoptera order. Answer is option A. Next one. During metabolic stress, which of the following amino acid generally dispensable in greater amount than can be readily synthesized in the body? So it is glycine. Answer is option B. Then next one. The precise arrangement of atoms of a protein can be determined by. So they can be determined by X-ray crystallography. Answer is option D. Then which one of the following is not naturally occurring nucleotide? So ATP, cGMP, CAMP, all these are naturally occurring, but DD-ATP, it is not naturally occurring. It is used for Sanger sequencing. Answer is option D. Then next one. The essential sites for recombination are known as chi sites. Answer is option A. All these are just direct question. That's why I am not explaining much. Then next one. The method used for prediction of 3D structure of a protein from known structure of related proteins is. It is called as homology modeling. Answer is option C. Then next one. Which protein is involved in the separation of two interlinked daughter chromosomes when DNA replication is terminated in E. coli? So it is topoisomerase 4. I think this question has been repeated for the same for PhD entrance examination. I think for biochemistry subject they have asked somewhat a similar question. So please refer those question papers also if you are attempting only the biotechnology section. I keep answering as many question papers as possible so that you can know how far you know the concept in what your subject, and also you will get to know more questions being multiple choice questions answered. You will get more practice by answering multiple choice questions. Then next one. How would you expect DNA methylation to alter gene expression? So DNA methylation, it decreases expression. Answer is option D. Then next one. In an alpha helix, the carbonyl oxygen hydrogen bonds to the amide nitrogen to stabilize the alpha helix structure. Therefore, the shortest stable alpha helix that can be detected by measuring circular dichorism is. So alpha helix, they told that it is stabilized by hydrogen bond between an amide hydrogen of one amino acid and a carbonyl oxygen of another amino acid, which is 4 amino acid residues away. So the shortest stable alpha helix that can be detected by measuring circular dichorism is 5 amino acids long. Answer is option B. Then next one. In southern blotting, nucleic acid on the nitrocellulose membrane is denatured by treatment with NaOH. However, in northern blotting, this is not done so because the nucleic acid get hydrolyzed. Which chemical nature of the nucleic acid in northern is responsible for this? So, northern is used for detecting specific RNA molecules from among a mixture of RNA, and whereas southern it is for detecting DNA. Now the RNA, it has got a 2' OH group, and this hydroxyl groups makes RNA less stable than DNA because it is more susceptible to hydrolysis. So the answer here is option C, the presence of 2' hydroxyl group. Then next one. Common step for life cycles of all viruses is, it is mRNA synthesis. Answer is option B. Then PD1 and CTLA4. That is option C, that is Tasuko Honjo and James P. Allison shared Nobel Prize for their work on PD1 CTL4. That is the answer. Then mating type silencing in yeast is regulated by expression of mating pheromones, temperature, expression of histone deacetylase, all of the above. So that's it. Please do like, share and subscribe, and please keep watching all the previous year question papers. I have already uploaded so many. You can refer them also to improve your MCQ skills. Thank you.